the next keynote speaker we're going to hear from is Ian Wilson, and we'll be hearing more about how his research has involved both user facilities and the kind of research he's been involved in. Thank you, Linda. I know as we segue over here, I'm going to hand off the introduction of Ian to my board director, Michael Becker, who's a scientist who works at Argonne National Laboratory. And Michael knows much more about the nuances of Ian's skills and background. So I will let him do the sort of introduction here in a way that will be a bit more fluid than I think I might be able to reference Dr. Wilson's research. Michael, over to you.
Thank you, Dan. It's my pleasure to introduce Ian. So Ian Wilson is the Hansen Professor of Structural Biology and the Chair of the Department of Integrative, Structural, and Computational Biology at the Scripps Research Institute. His group uses structural biology and biophysical techniques to investigate how the immune system combats infectious diseases caused by microbial pathogens such as influenza, HIV, and SARS-CoV-2. Many of these pathogens are zoonotic, which means they can go between non-human animals and animals. And I think from the title of his talk, he's going to emphasize mainly the effects on humans directly. I'll just have a little aside. The effects on animals also can be very large. If we take avian influenza, for example, there was an outbreak in the US in 2022 where greater than 40 million chickens were lost. So you notice that in your egg prices, and the cost of the economy was greater than $3 billion. And this is not even mentioning what goes on with the wild birds out in nature. So back to the human focus. And I'm going to mention that Ian is a giant in the field, and he has authored over 875 papers. And I have to read this. He's a fellow of the Royal Society of London, a fellow of the Royal Society of Edinburgh, which I hope I pronounced properly, a member of the American Academy of Arts and Sciences, international member of the US National Academy of Sciences, and an honorary fellow of the Corpus Christi College, Oxford. He was also the director of the Joint Center for Structural Genomics for 16 years, which helped to pioneer high throughput methods for structure determination, like Linda mentioned, for X-ray crystallography and NMR, which resulted in 1,600 novel structures deposited to the protein data bank. So Ian's work has been crucial for understanding pathogenesis on a molecular scale and for applications such as vaccine design. Also, user facilities such as synchrotrons help to enable Ian's work. And so without Ian's work and folks who are working on these types of problems, as well as the synchrotrons and cryo-EM facilities and NMR and so on, without these two components, we would truly be in the dark ages. Ian, could you please enlighten us on the future? Thank you.
Thank you very much, Michael, for that very kind introduction. And thank you for the kind invitation to actually talk to you here today. So I'm a structural biologist and I work in infectious disease. And so what we're trying to do is to understand how viruses and other pathogens infect us and how our immune system responds. And so the sort of main pathogens that we work on in my lab are influenza virus, HIV, hepatitis C virus, SARS-CoV-2 and related coronaviruses, as well as the parasite, the malaria parasite, the Plasmodium falciparum. But I'll focus today on the viruses. So what we're trying to do is to sort of basically use the information that we get from how the immune system combats or tries to combat these viruses to try to design better or new vaccines and better therapeutics such as antibodies. For influenza virus, we know that we've got the seasonal virus, a seasonal virus that we get, and we can get immunized against it every year. In fact, I'll be going to get my shot today. But it would be nice if we could actually get a more universal vaccine that could actually protect ourselves not against future outbreaks of these seasonal viruses, but potentially against pandemic viruses. For HIV, we don't have a vaccine at all. It's been a long process. There is progress that's getting made, and quite exciting progress actually, and I'll talk a little bit about that. Hepatitis C virus, we don't have a vaccine for that. We have good effective drugs, but again, it would be good, especially to prevent reinfection, to be able to have a vaccine. We've all gone through SARS-CoV-2 and we know what the values are of a vaccine. Can we use this information now to actually think about getting not only the SARS-CoV-2 and all of the Omicron and future variants, but can we think of trying to protect against SARS-3 by the information that were derived? For malaria, there's a vaccine out there called RTSS, and there's been a sort of a repackaged version of that. There's very exciting results have just come out recently at R21, which gives about 50 to 75% efficacy. And then again, can we use this sort of structural information from how our immune system is interacting with this particular parasite to actually improve upon that vaccine? So as you heard about in the previous talk from Linda, then we've got an amazing national facilities, and we've got these synchrotrons, and we use all of them in our work. We couldn't do our work without all of these facilities. So we've got SLAC, SSRL, Argonne, the APS, Berkeley Advanced Light Source, Brookhaven, and the National Light Source. And we sort of go to all of these to collect data. These facilities have got better and better over the years. They've got brighter and brighter. When I first started doing X-ray crystallography, it used to take me a month to collect a data set in the lab. We can now collect that in a few seconds with smaller and smaller crystals, and we can do more sophisticated experiments that we could ever do in the past. This requires a lot of different agencies. We heard about the Department of Energy funding these national facilities. At NIH have also beamlines at APS, and they also are providing the support for our research. And so without that, we couldn't obviously do the work. We're also supported by USAID. We have collaborations with people that are supported by the Department of Defense. And then for non-government, Bill and Melinda Gates Foundation has been very important. And for the vaccine design, IAVI, and for malaria, PATH. So getting on to these viral antigens, then they're all trimeric. They all appear. These are the so-called spike proteins that we heard about with coronavirus, the so-called S protein or spike protein. These are all the different spike proteins from these different viruses, from Lassa, from Ebola, influenza, HIV, coronavirus, and from Nipah. So they're all trimeric proteins. So there's 3 copies of each of the subunits. And they're cloaked in these blue things, and the blue is the carbohydrates. And that makes it very much more difficult for the immune system to be able to actually recognize these and mount a very good immune response. So part of what the immune system does is it tries to sort of, if we take HIV here, it tries to get in to these spaces that are not covered by carbohydrate and attach to them. But with something like HIV, for example, there's 81 glycans on this trimeric protein. And so there's very little surface that's available. And so that's really how the viruses help protect themselves from our immune system. When we look at the distribution of these spike proteins on the surface, then we can see that some of the viruses differ quite a bit. For HIV, there's not very many spike proteins on the surface of the virus. The virus is in blue here, and it's got a membrane, and all of these are some envelope proteins. They've all got a membrane with a spike protein protruding from the surface. And we can see that there's a rather dense rather paucity of envelope proteins on the surface, which again makes it more difficult for the immune system to recognize. For SARS-CoV-2, there's a few more, and they have different conformations, and we'll sort of go into that later, but they sort of have part of the domain, the receptor binding domain, can go up and down, and so that can protect, again, a vulnerable epitope, but it needs to be up to engage with receptors, so it must be in sort of this flickering type of form. And when we're thinking about vaccines, what we're trying to avoid is these sort of elongated structures here, because these are the post-fusion structures. So all of these viral antigens have to change their conformation. They bind to a receptor on the surface of our cells, and then basically they have to then penetrate through our membranes. So they have a sort of membrane fusion activities. And so these spike proteins are involved in that. And to do that, they have to change from this, what we call pre-fusion conformation, to this post-fusion conformation. And this is not the conformation that we want to see in a vaccine. And so sometimes stabilizations of proteins have to go on to keep it in this pre-fusion form, as I'll show you for SARS-CoV-2 just in a second. For influenza virus, you can see there's a much higher density of these spike proteins on the surface. In fact, there's two different spike proteins. There's the hemagglutinin that helps you get into cells. It has the receptor binding site on it, and it binds to sialic acids on the surfaces of our cells. The yellow one here is neuraminidase, and it's a receptor destroying enzyme, and it cleaves the sialic acid off the infected cell and enables these new progeny variants to escape and infect new cells. So if we go, I'll go through in the order of which I started working on things, going through influenza, then HIV, and SARS-CoV-2, to show you how the spike protein structures were actually determined and what they look like. So we have to go all the way back to 1975, where Don Wiley and John Scahill produced these crystals of the hemagglutinin glycoprotein from influenza virus. In fact, what John Scahill did is he just basically grew up the virus in chicken eggs and cleaved off the hemagglutinin with an enzyme called bromelain, and basically then purified out the hemagglutinin, and that and purified it in sufficient quantities to enable crystals to be formed. These crystals diffracted, so they started this project. And the same year, I actually went to Don's lab and started working on the structure. And a few years later, in 1981, we actually got the structure of this. So this, as you can see, we didn't have all the fancy graphics that we have nowadays. All of these are hand-drawn. We didn't have any other programs. And so this was a depiction of the trimer, and this was a depiction of one of the three monomers of this trimer. And what we could do then, we didn't have nucleic acid sequencing, we had protein sequencing. And if we looked at the evolution of this virus from the pandemic in 1968 through to 1977, and then just put these red dots or red stickers on this cartoon of the hemagglutinin, one could get an indication where there were mutations going of where the antibodies were actually likely to be binding with the so-called epitopes. And so one could see that most of those tended to be on this head domain in and around the receptor binding site, which is here, and not on the lower half, which is much more conserved, which contains the fusion machinery. So fast forward a few years, we've done a lot of different structures of the hemagglutinins at this level of resolution. They all look very similar to one another, but their surfaces are all different. They all have different amino acids. So we looked at, for example, the 1918 pandemic HA. Then that was followed by an H2N2. This was an H1N1 virus. This is the hemagglutinin 1 neuraminidase 1. H2N2, the hemagglutinin neuraminidase changed. And then this pandemic, and the structure I showed you previously, it was an H3N2. Then the next pandemic we got was quite a number of years later with this the so-called swine flu pandemic, which was another H1N1. Well, what we've been concerned about are all these so-called bird flus, especially starting with H5N1 and going on to things like H7 and H9. And so all of these structures look very similar to one another, but their surfaces are all different. So antibodies that we made our immune system recognizing one doesn't necessarily recognize another virus. And so these can be divided up into different groups. So for influenza A viruses, which are the ones that caused us the most severe disease and deaths, they can be divided into two groups. There's 18 different subtypes, 16 of them appear in wild birds, 2 in bats. And they can be divided into two groups, with the majority of them being in group 1 and still quite a number in group 2. So what we'd like to do is to get antibodies or a vaccine that would be able to effectively combat as many of these different subtypes of influenza A virus as possible. So then if we go on to the HIV envelope trimer, we waited quite a number of years from 81 to 2013 to get this. This was a very hard structure to determine. It required stabilizing mutations that were put in by John Moore's group, by Rogier Sanders and James Binley when they were graduate students. And so, oh, sorry, this is jumping. So what we can see is that we got not only a crystal structure back in 2013, but we got an EM structure. So this was start of the so-called, just prior to the EM resolution revolution. And so the EM structure was done in Andrew Ward's lab, and Jean-Philippe Julien, postdoc, solved the structure in my lab. And so we could see then the trimer, and we actually had antibodies bound, so we could actually see where some of the epitopes were. So again, that sort of opened the door to be able to do more structures. And so in the next 3 years, a number of structures were done. These were all crystal structures at the time. Most of them were crystal structures, and then EM structures started to come in as well from Andrew's lab and also from other labs. So we can see that these structures, they all have the same sort of general topology. And on the right-hand side, what I'm showing is basically all of the glycans. So these are different types of glycans. You can have sort of high mannose glycans, which are sort of the immature glycans, and then you have ones which are complex, which are much more decorated with other sugars. And so if you look at the top surface of that in the center picture there, then you can see that there's very little space there. The gray is the protein surface below. And so the antibodies have actually got to get through these spaces to be able to contact the surface below. So this is really quite a challenge for our immune system recognizing HIV. For coronaviruses, the first coronavirus structure was determined actually in 2016 in a collaboration between Jason McClellan and Andrew Ward. And this is also a trimer structure, and this was done by electron microscopy. And so they wanted to go on and look at MERS and SARS-1, the original SARS virus. And when they actually looked at it, this is negative stain EM here. When they looked at MERS then and they expressed it, you could see that there's a lot of these long elongated spike structures. This is the post-fusion conformation, where you really want all of these type of ones, which is the pre-fusion. That was the same for SARS-2, where we got a lot of this. So in order to stabilize it, then similar to what one did with HIV, one considered proline mutations. And Andrew and Jason, they designed, in their labs, designed these 2p mutations at the top of one of the helices, which actually increased the yield of the protein, but also increased its stability. So when you now looked at negative stain EM, you could see that they were all these nice uniform trimers. So we didn't know the SARS-2 pandemic was coming. And so these 2p mutations turned out to be extremely valuable for that. So in all of the vaccines, whether the mRNA vaccines, which were a great breakthrough at the time, and we got awarded the Nobel Prize this year, and for that technology. And so all of these constructs from Moderna, from Pfizer, J&J, a protein construct from Novavax, used these 2p mutations to help stabilize that, enable our immune system to recognize the correct form of the spike protein in order to help combat the SARS-CoV-2. So the first structure of a SARS-CoV-2 spike protein came out really very quickly after the initial sequence. It was in a matter of just a few weeks. This was from Jason McClellan's lab. David Wiesler produced one shortly thereafter. Again, this was an EM structure. And what one can see in the middle here is the various sort of domains. There's the top component, the RBD is the receptor binding domain that binds the ACE2 receptor. The NTD is the first domain. It goes into the RBD, and then these connecting domains to the S2, which is the fusion machinery. So I've only got one of the trimer components, one of the monomers is highlighted here. The other two are in gray. And this is the form that we don't want. This is the post-fusion form. So this is the form that we want in the vaccine when it's delivered. So I've talked to you so far about how the structures of these antigens on these viruses. This is very important information to know what to target. But what about how the immune system recognizes it? So this can be a fairly long process. And so if I start with the flu hemagglutinin down at the bottom here, then from that first structure and these red dots that I showed you, we had an idea of where the antibodies were targeting. And so we called these basically, so we just called these A, B, C, and D sites. But as we went on, we sort of started to find other sites, but it wasn't until 2008 when it was possible to isolate single B cells, and this was done in Michael Nussensweig's lab, Dennis Burton's lab, and other labs. And so the ability to do that enabled us to produce far more human antibodies against particular targets that we knew the specificity for and the sequence for. And so you can see then rapidly some other epitopes started to appear. So it's really, even up until last year, new epitopes have been found. So this discovery process is quite long. But why is this important? It's important to identify these so-called vulnerable sites that the immune system can target. And these are the sites that we want to target with a vaccine or with therapeutic antibodies. So identification of these sites from from the structural work and with all of the antibody work is sort of critical in the vaccine and therapeutic design. The same is true for the HIV envelope protein. In 1992, we knew about the receptor binding site, the CD4 binding site. And we started to know about antibodies right down near the membrane, antibodies that actually interacted with carbohydrates. This was very unusual, but because of the density, it was perhaps not surprising. And then again, from 2009 onwards, a whole lot of new epitopes started to be discovered. So let's look at these epitopes in a little bit more detail and show how that information was of some value. So for influenza hemagglutinin, here is the trimeric spike. And one of the first antibodies, there were actually 3 groups that produced very similar antibodies at the same time. We worked with Crucell in the Netherlands. That's why this is called CR. 6261 is the name of the antibody. And much to our surprise at the time, we were looking for broadly neutralizing antibodies. And we were expecting that basically most of the antibodies would be against the head, but that's hypervariable. So not surprisingly, the antibodies were pulled out, targeted the more conserved stem region. Antonio Lanzavecchia produced an antibody, so that this first antibody was against 10 of the 16 avian subtypes. And so that really hit 2 of the human pandemic viruses. Antonio Lanzavecchia found ones that hit all of influenza A viruses. So this, from having no human broadly neutralizing antibodies, in a couple of years, we had really extremely broad ones. And then, again, in a collaboration that we had with Crucell, they pulled out an antibody that could actually bind to influenza A and influenza B. And you can see they're targeting a very similar site. And then VRC and later showed that they could actually induce antibodies against that site with a vaccine, and this targeted the same site as these antibodies above. And what was interesting is that if you look at the tips of the antibodies, and you look at the amino acids that are actually making the most of the contact, these are the amino acids that are depicted here that are in the tips of these so-called complementarity determining loops, the binding loops of the antibody. And you can see that there's 5 pockets being filled. This one here is almost always an isoleucine, this is a phenylalanine, this one's a tyrosine. So that then gave us a lot of good information for thinking about targeting not only with antibodies, with vaccines, but as we'll see with small molecules. Well, was there any hope of a low breadth against the head? And the answer to that was yes, because blocking receptor binding is really makes the antibodies really much more potent. And so a number of antibodies that were found that had some breadth that weren't as broad as the ones to the stem, but they had some breadth against particular subtypes. And so we can see there's a number of antibodies bound here. Obviously, they can't all bind at the same time. But what they did is that they put one of their 6 binding loops, inserted it into the receptor binding site. And so here's a couple of different antibodies showing that. And here's all of these different antibodies showing they're all putting one loop in there. And what they did was something very interesting, is that the tips of the loops here, there was an aspartic acid. And this aspartic acid mimicked the receptor. The receptor is sialic acid, and here's the carboxyl group of the sialic acid. Half of the antibodies also did another sort of mimicry. They put this aromatic residue that mimicked this acetamido side chain, which stacked on top of a tryptophan below. So really what the antibody has to do is to try to put a loop in, try not to touch the hypervariable sides around, and so almost having like a one-loop antibody. In fact, CO5 is a one-loop antibody. It only makes contact really with this one loop. And so basically to target this very small binding site for where sialic acid binds. So how could we use this information then to try to think of getting better vaccines and to go towards a more universal vaccine? So one of the simplest things to think of was that we don't want to target this hypervariable head. So if we could chop this head off and have a so-called headless HA, that could actually be a good starting point for a vaccine. This turned out to be a little bit more complicated to actually produce than one could have thought. It has actually 3 different chains, because there's 2 chains here, the blue chain and the green chain, and the blue chain goes up and then it comes back down. So that means that there's two parts blue and then this green part. So again, with Crucell, they helped design a headless HA And so this is it shown here. I did the crystal structure of it. And we put some extra helices on here to help keep it trimeric. And we could show that these antibodies that we saw in these previous slides that were actually broadly neutralizing could actually bind to this construct. On the same day as our paper came out, the paper came out from Barney Graham's lab at the VRC, and they had designed a slightly different construct. The essence of it was the same. They were still trying to get this headless HA, just doing some different connecting helices. So there were two new possible vaccine candidates. So since then, there's been other ways of thinking of doing that. So, for example, you could actually take these headless HAs, as we saw previously, and put it on a nanoparticle. So when you increase the density there, you actually tend to increase the immunogenicity, and that the immune system responds better by putting more of them on a particle. For the receptor binding domain, we know there's a lot of variability, but what if you put a lot of different receptor binding domains from different viruses on a nanoparticle? Can you get the immune system to basically focusing only on the conserved regions and use that as a vaccine? Another way you could do it is to sort of then, rather than doing the headless HA, is basically shield this hypervariable surface by putting more glycans on there. So just basically coat the surface in glycans and prevent antibodies from recognizing it and focusing the immune system down on the bottom part. Then Peter Palese and Florian Kramer also wanted to try to get a vaccine response against the stem here, the highly conserved stem. So they thought, well, if we put different heads on there and heads that we had never seen before, then we can actually maybe elicit more antibodies to the stem by priming and boosting, and the only conserved part would be the stem. And in fact, that's what they did. So, and if you think that we've got pre-existing immunity, we've got some to the head that will vary from season to season, from virus to virus, but we'll have some, albeit a lot less of a response against the stem. Can we boost this response by putting a different head on there and then changing the head and boosting again, and basically ending up with those types of antibodies? At a much higher concentration of those antibodies against the stem. And in fact, that appears to be what is happening. And so they've done this for both these group 1 influenza A viruses and group 2. And so again, with crystal structures, with data collected at the synchrotrons, we've shown that basically these can actually form trimers with these odd heads. They have a little bit more separation at the bottom. But if we look at the crystal structure, then again, neutralizing antibodies can bind to this. So this particular construct can work quite well as an immunogen. And so it compares to, so this is the H5 structure, this is the H1 natural structure, and this is this artificial chimeric type structure. And so that's used as an immunogen to focus the response against the stem. And in fact, we've got antibodies back from that, from Jenna Guttmüller, Patrick Wilson, Florian Kramer. And basically, yes, you can elicit antibodies, and these antibodies are binding in exactly the right place to the stem, so showing that that type of design can actually work for a vaccine. I put in the title talking about therapeutics, and so when we worked in the structures of the first antibodies to that first antibody to the hemagglutinin stem, David Baker at the University of Washington basically got interested. Could he design something that was much smaller than an antibody? And so he designed this sort of scaffold of, you can see it's an alpha helical scaffold. And then he attached basically side chains to that scaffold that could fit into these pockets that I showed you previously. So he tried to fill 4 of these pockets. And in fact, all of these early structures that were done by Damien Eckert in the lab, and in this collaboration with David's lab, we compared the crystal structure with the actual design structure. And you can see the design was almost perfect, even to the rotamers of the side chains. He's gone on and tried to make, with Aaron Chevalier and this group, to try to make an even better design and much more potent. And he's found now small proteins that are much better than the most effective of antibodies as sub-nanomolar neutralizing capabilities. So we thought, well, okay, if you can go out and design a small protein, what about designing a peptide? So we looked at different antibodies. These are two different antibodies. This is the one from Antonio. This is the one that we worked on with Crucell. And if we're going to design a cyclic peptide, we want sort of a more continuous stretch, and so of amino acids. So this looked like a pretty good template. This is the CDR3 loop, one of the binding loops of the antibody. It basically forms most of the interactions with the stem here. We also wanted to include this area here. So this was our target design. Having this is the starting point. Can we pick up this interaction? And then by cyclizing it with proteogenic amino acids, then we actually made this compound. Again, this was made at Crucell. We solved the crystal structure, and this turned out to be quite potent. And so this also prevented the fusion activity of the virus. Well, could we go even smaller? And so we could use this information either from the small proteins or for the peptides, and we could basically use that to compete with a library of small molecules. And we would then know that from this library, if we actually basically competed with a small protein or a peptide in this, we knew that the small molecule was targeting the right area. It was previously thought that the hemagglutinin was undruggable, and lots of people had tried. This would seem to be even a more particularly difficult surface. It's very hydrophobic and it's very flat. But from this library, again, in collaboration with Crucell, we pulled out this particular molecule, and we can see that it bound in the same place as the antibodies. So I put in green here where the antibody side chains are filling on top of the small molecule, and it's binding in an identical place. And so you could give this to mice, and basically you could completely protect in mice with this small molecule. So this was pretty remarkable, actually. If you'd asked a chemist you could do this, they would have thought probably not. And so what we're seeing is that we started off with this structure. David Baker's group kept on working to smaller and smaller proteins. So we go down from around 50 kilodaltons to 10 to 5. Then we had this cyclic peptide, and then we have small molecule. And then with Dennis Wallen, Seiya Kitamura, we've actually been working and improving that to get even smaller and smaller and more potent molecules that can inhibit that. So this would be a really good place to target because you want to stop the virus getting into the cell. I've talked mainly about the hemagglutinin, but the neuraminidase has sort of been so almost forgotten in a sense from the vaccine perspective. It's not been forgotten from the drug design point of view, because Tamiflu and Relenza are compounds that one can take to try to ameliorate against infection. They don't actually prevent the virus getting in, but they prevent basically the virus spreading. So with two collaborations with Jim Kroll and then with Ali Elibadi and Florian Kramer, we've looked at antibodies to the neuraminidase. Neuraminidase is different from these others. It's a tetramer. And here's antibodies binding around the outside. And we see this sort of molecular, mimicry of the receptor going on in these antibodies as well. Because again, to get into the binding site, they throw in a long CDR loop. And at the tip of this loop is a glutamic acid, which actually, again, mimics the carboxyl and the sialic acid. These antibodies were specific for H7N9, one particular subtype. Ali and Florian pulled out antibodies which had remarkable breadth. And they actually pulled out one antibody that could basically neutralize all of influenza A and influenza B. And again, it threw this, this long CDR loop into the binding site. So this has come into the forefront again as a possible target, not only having sort of hemagglutinin in the vaccine, but also neuraminidase to try to sort of get more potent and universal vaccines. For HIV now, if I go into that, then I can show, I'll show you here how difficult it is to actually bind antibodies to bind to HIV. And so I've got a number of different binding sites here, all the different epitopes that have been found over the years. And these sort of yellow things here, green things here are carbohydrates. So you can see that basically you've got to get so the side chains of the antibody that actually sort of penetrate through these carbohydrates in order to bind to the HIV envelope protein. So we've sort of defined pretty much all of these sort of different epitopes now, either by X-ray or by EM. And so there's also another interesting thing, not only carbohydrates play a role in these antibodies, but when you're trying to target right down at the base of the spike protein where it enters the lipid membrane of the virus, there's an epitope all the way down here, and then that antibody can also interact with lipids, and so it started to acquire some specificity for lipids. So there's a lot of complicated things going on with HIV. These antibodies also are extremely highly mutated, and it takes often like 2 years to be able to develop some antibody with neutralization breadth. So that tells you if it takes 2 years in our bodies, how are we going to do this with a vaccine? How many shots are we going to need to be able to get such a response? But the good news is that as we've gone on over the years, so prior to 2008, which I told you there was this sort of revolution in the way in which B cells could be isolated, then we had a number of different antibodies which were basically not very potent. Some of them had extreme breadth, particularly ones down to the stem region here. But over the years, what we've been looking for are antibodies, pulling out antibodies that are very potent and have breadth. So we want to have antibodies in this quadrant over here with high potency and high breadth. And so these are the antibodies that we can actually use to define the epitopes that we're going to target with vaccines or with therapeutic antibodies. So if we take one antibody, one example of antibodies in this region here, all these blue ones are ones that actually target the receptor binding site. And so the first antibodies against the receptor binding site were weakly neutralizing with limited breadth. And then we got on to ones which were modestly neutralizing with a 30% breadth against a panel of different HIV viruses. And then we got up to VRC01, which was much more potent at higher breadth, and up to today where we've got one which has almost got breadth of 100%. So this is defining then a target that we'd like to have in a vaccine. And so when do you keep stopping? At this stage, it was thought it was promising to actually think of using this as a target and to try to focus the immune system against this particular site. And that's what, in fact, what Bill Schiff did. He, instead of having this sort of big protein, he basically figured out how to sort of make a smaller version of that the antibody would bind to. And so he made what we called an engineered outer domain. And this bound to these same antibodies. And these antibodies were all from one particular germline family. And so they were from a family, there's about 50 different heavy chains in our immune repertoire. And this is what was one called VH12. So what Bill did was he basically took this and he put it on a nanoparticle. So there are many copies of it, 60 copies of it injected it into people. This is a human clinical trial conducted, it was called the IAVI G001 trial with an adjuvant. And so basically what one was looking at was trying to see what sort of antibodies came out and where are they from this particular family. And in fact, that turned out to be the case. Whether you use 20 micrograms or 100 micrograms, pretty much everybody produced these VRCO1 type antibodies in this family. And so we've looked at some of these antibodies that were pulled out from this immunization. And in fact, here's the original antibody here. Sorry, got a little squashed in what I was actually just trying to reduce the file size. But you can see that all of these antibodies were pulled out, and they're all very similar to this particular antibody. This antibody can pair with different light chains in a natural response, in a response to natural infection. In fact, all these different light chains are also represented in this vaccine immunogen. So this is not the solution, but this is basically a promising start to show that you can actually start the immune system in the right direction by pulling out the right types of antibodies that you know that neutralize the right sort of family. And now the next question is, how can you mature those to be able to get something that would be neutralizing that could go into a vaccine? For the last example, let me talk a little bit about coronaviruses. And there are different types of coronaviruses, alpha, beta, gamma, and delta. And the ones that we have to worry about for self-human infection are alpha coronaviruses. There's two seasonal ones in this class. And then this larger class of beta coronaviruses, where we've got two seasonal ones, SARS-1, MERS, and SARS-2. And we know that these viruses are transmitted initially from bats through different species, camels, pangolins, civets, raccoon dogs, bamboo rats, etc. And so this is the sort of family that one is looking at, that we saw the structures of these spike proteins from these earlier on. So at the start of the pandemic, we didn't have any antibodies against SARS-CoV-2. So Mong Yuan and Nicholas Wu in the lab set about trying to express the receptor binding domain, but at the same time looking to see, well, what other antibodies are out there. And so they looked at some antibodies to SARS-1 from the infection some 20 years earlier. And so there were a number of antibodies that one could actually pull out, express. And one of them, this again from Purcell, actually bound well to SARS-1 and moderately well to SARS-2. So this gives us a basis for then trying to grow crystals with the receptor binding domain of the spike protein along with this particular antibody. So we got crystals very early on, and it was really incredibly fortunate that the synchrotrons remained open. And so we actually immediately, when we grew these crystals, we sent them to the APS and basically collected data. Here's Mong processing the data, solving the structure. And we actually solved that structure. So here's the receptor binding domain from SARS-CoV-2 with the antibody. And by definition, because of the way it was pulled out of cross-reactive between SARS-1 and SARS-2, it was a pretty broadly neutralizing antibody. And this bound to a highly conserved surface that was only 4 changes between it and between SARS-1 and SARS-2. And this surface was much more highly conserved than the receptor binding site, for example, which was only half of the residues are concerned, even fewer now with all of the Omicron mutations. Whereas this is an extremely highly conserved site, and that provided the first really good target for thinking we would go up for a broad neutralizing response against SARS-CoV-2. I wish everything could move as quickly as it did then by collecting the data. We collected the data on March the 6th. We submitted the paper on March the 14th. It got accepted on April the 1st. It appeared online on the 3rd. So everybody was moving really fast in those days. and everybody was cooperating together to get this information out. So this antibody, like the receptor, can only bind when the RBD is in the up conformation. And so you can see when it's down, you can't see the epitope. Here's the epitope. And so this flips up in these other representations here. The RBDs are down, just to sort of highlight this up conformation. So we didn't have to wait. The whole world started working on this, and lots of antibodies started to appear. And so here's a lot of papers that appeared in the sort of spring time to early summer of 2020. And we collaborated with a number of different groups, including Dennis Burton's group here at Scripps, Rogier Sanders and Marie van Gils in the Netherlands, and a number of other different groups. So from having sort of no SARS-2 neutralizing antibodies with a whole plethora of them, and so we could sort of group those and look at them and look and see how the immune response was to this particular pathogen. And so here are the numbers of, here's all the different possible heavy chains that we could have from our repertoire in our immune system. And we can see it was dominated by a few up here, and then this long tail, and some were never used at all. So what was perhaps not as surprising is that there was not a lot of somatic mutations. So this is usually the process you undergo to increase the affinity, because these were pulled out from fairly, from people who were early infected, and the antibodies were pulled out fairly close after their infection. But what was surprising was how potent these antibodies were. So we had antibodies that basically had almost no somatic mutations that were extremely potent in the nanograms per mil range. So that says that our immune system has sort of almost off-the-shelf antibodies ready to go that could actually interact with SARS-CoV-2. And I think we were very fortunate in that regard. So we had to look at the most prevalent one of these to see where it bound. And so in this collaboration with Dennis Burton's group, we actually collected data up at SSRL, synchrotron there. And so we looked at two different antibodies, and basically these antibodies bound in the same place. And you could see that if we compared it to the receptor binding domain interacting with the receptor, basically these antibodies would inhibit receptor binding site. They bound with nanomolar affinity and with high neutralizing potential of the capability of nanograms per mil. So lots of antibody structures started to appear, and we started to sort of categorize them into groups to see where the most prevalent were ones that where they were binding. And so most of the antibodies bound to the receptor binding site. And then this is this for this first antibody that I showed you, the CR302, there was a patch down here, another lateral patch on the other side. So these patches here, the antibodies that bound to these patches were cross-neutralizing, where these tend to be far more specific and were more subject, as we'll see, to the mutations that were acquired in SARS-CoV-2. And so when we actually looked at these antibodies, then basically we could subdivide them into 4 areas. And we could see, again, I sort of messed up my slides here this morning when I actually tried to compress it. So, but we can see that these antibodies on the top are all binding to the same place, and the antibodies to the bottom are binding to a different place. So this is sort of given a spectrum of the A and B sites were actually the most prevalent sites that were being pulled out for binding. And if we looked at, they've then looked at that with regard to the types of heavy chain genes, we can see this whole class at the top was dominated by 353, and then these other, we had 353 VH12s, VH169s, VH330s. These were the top 4 antibody categories of the ones that were produced most predominantly with the SARS-CoV-2 infection. So I show that because then that's a worry then, because when we start to get mutations, can you knock out whole classes of antibodies with relatively few mutations? And we'll come to that. So one of the more remarkable things that's happened is that rather than having to worry about people getting tests, is the wastewater, and you can test what's going on in what the prevalence is. So we've seen all these different waves. There's, I don't know how many waves there've been now, but there've been a large number, far more than we've seen for any other pandemic. And so here's the different waves of Omicron that are being detected right up to the most recent ones in the San Diego wastewater. If we look at this, these different mutations, we can see that it was kind of amusing at the outset. We thought that there was going to be very few mutations, that this wasn't going to mute, that SARS-CoV-2 wasn't going to mutate very much. At the outset, it didn't seem to mutate too much, but then we got these huge waves of mutations in Omicron. And they're largely associated with the receptor binding domain or with the NTD. There are certain mutations that appear frequently, and these are highlighted in red there. And so these are the ones that we looked at to see what is their effect on these different classes of antibodies. And so when we look at these different classes again, this was as a more update. You can see even with more sequences available, you can see how that more of half of the response is dominated by these 5 antibodies. It's actually 4 because 366 is pretty much the same. It's only 1 amino acid different from 353. So we now then look at 353 and VH12 and where the mutations are that we started to see in these variants. Then we can see that for these 353 antibodies, the RBD is in this grayish color, yet whitish gray color. Then this 417 mutation would knock out those class of antibodies. There was actually a different binding mode for this, and I don't have time to get into it. That was affected by the other. So this was a double knockout of this particular class. For VH12, then it was, again, if we have the RBDs all oriented the same way, the antibodies at the top here, then we can see 484 knocks out this sort of class of mutations. And we can see from the details at the bottom, we understand why. If you change the charge on that from a glutamic acid to a lysine, that's going to knock out these particular interactions. So for the RBD, only the lateral patches in the early days, all the way up to, from alpha to delta, there's been some mutations started to emerge now in these particular patches, but very few compared to the receptor binding site. So there is some of these antibodies actually have a considerable breadth compared to the ones to the head. But that explains why all of these antibodies that were actually produced and that people were being given, they basically have all been escaped now. And so one has to look for more areas where are other areas that one could target with antibodies that have greater breadth, where mutations don't seem to emerge. And one of these areas is down in the stem. So we go back to the stem again. This is really a recurring feature for all of these. This is where all of the conservation is happening. And there's a peptide down here, and just before this would enter the membrane, that is highly conserved in beta coronaviruses and going all the way through bats and human and MERS viruses. And it's very different in alpha coronaviruses. So in these antibodies from Dennis Mertens, I have this particular antibody, neutralizes SARS-1 and SARS-2, and many of these other viruses. And so Andrew Ward's lab showed, in fact, this particular antibody, it bound right down at the base here. So this is a false color. This is the antibody here down at the base. And here's one of these peptides. We actually had it synthesized. We did a crystal structure, and we can see that it forms, it sits in a nice group between the height and the light and the heavy chain. And so here's the update on that. There's a lot of different antibodies that have been pulled out against this region. They tend to be restricted to particular families. They're not as potent, but they're extremely broad. And the potency may have to do with because it requires this to open up in order to get the antibodies in. And then the last episode I'll show you is one that was pulled out by Joshua Tan at NIH and NIAID. And this is against a conserved fusion peptide, which binds to alpha, beta, gamma, and delta coronaviruses. So some of these antibodies are extremely broad. And again, we've done crystal structures showing the peptide binding in the groove, the fusion peptide, and a number of other different antibodies is targeting this particular region. So we've only had, I showed you, like in some cases, 40 years of finding antibodies. We've only really had 3. But we're now, as you can see, we're populating this area. This is the quadrant we want: potent neutralization, broad reactivity. Most of the antibodies have been to the RBD, some to the NTD, but there's large swaths of the surface that have been largely unexplored where there might be greater breadth. So now is not the time to stop. This is where we should be going on, trying to sort of look and try to figure out, can we actually then target these more conserved regions and prevent SARS-CoV-2. So these are the antibodies that we determined. And so these, again, thanks to all of the synchrotrons, we could not have done this if they had not remained open. We've done a lot of different antibody structures and antibody structures to the RBD, to the stem, and to the fusion peptide. And we set up this a number of years ago, this vaccine pipeline. We don't, in our lab, do any of the antibodies. We're highly dependent on these fantastic collaborators that you have that produce these exciting antibodies. We do the structural biology. We work with groups like Bill Sheaf here and other people who are designing vaccines and therapeutics in order to push all this way into the clinic and hopefully get better universal vaccines and therapies. And without, again, we couldn't do that with all of the support from the agencies. So I'm not going to go through all of the people the individuals. I sort of not been able to do that at the time. I just want to show you that there's enormous teams of people that are required, not only the facilities and the funders, but large groups of people that are involved in identifying these antibodies and getting crystal structures. This is for SARS-CoV-2, this is for flu, and this is for HIV. So I'll stop there. Thanks.
Thank you, Ian. Incredible. This is so compelling to watch you go through the detail and to recognize the amount of precision and just puzzle solving that goes on in the discoveries that have been made here. I'm curious if you mentioned, of course, the different synchrotrons and the user facilities that are fortunately still available to leverage. Would there be a next particular project that you'll personally be involved in? Where might you be touching sort of base and interacting with user facilities that we could just get as a sense almost anecdotally of where you'll be connecting with user facilities? So I think, as you can see, that these projects go on for a long time. We're not finished with HIV. We're not finished with flu. Hopefully we're still going to get enough support to be continued to work with the coronaviruses, but there's lots of other viruses out there too. Mpox came and briefly went, but that'll probably come back. We've touched on the malaria parasite. There's lots of targets on that, and then there's a lot of other viruses which are NIH and others have been putting a lot of more effort into thinking of trying to tackle and try to get the vaccines. And so we've looked, there's a number of other emerging pathogens in those areas. So I think there's going to be more than enough to work on and more than us to keep us busy and going to the synchrotrons and collecting data and hopefully getting better vaccines and therapeutics for a number of different viruses and other parasites. Wow. Well, I'd say thank you for not only an objectively fascinating presentation, but more so the profound impact that this kind of research has on public health and just the security of humanity is kind of hard to almost put into words. Thank you.
It's my pleasure.
Okay,